The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
I don't want anybody to be feeling, super rushed. Not everyone will need the whole 2 hours. Some of you might, and that's okay. That's why I give 2 hours. If you finish the test early, just, have your phone with you or homework for another class or whatever. You can go kill some time somewhere because we will always have lab on exam days. So whatever time you finish the test, we'll regroup together in lab on Wednesday at noon like we always do. Any questions about what's coming up kind of today, Wednesday, into next week?
So for the pre-quiz today, we're doing
Yep.
6 and 8?
Yep, so you're going to have 2 pre-quizzes. The first one will be 3 questions from Lab 5. And then the second little half sheet of paper, I think I put 2 questions from Lab 6 and 1 question from Lab 8. And that was the one that would have been today. That's normal. I'm just added in. I didn't want to take away the Lab Exercise 5 because that would change the number of points for the class.
Yeah.
So, okay. Other questions? Okay, cool. So let's go ahead and get into Chapter 4. Chapter 4 represents a pretty substantial jump up for us if you kind of go back to that organizational diagram that we saw all the way back in Chapter 1. Remember, we started with atoms at the bottom. We then combined atoms to form a molecule, and that's where we were really for the last couple of chapters. Chapter 3, of course, was looking at the organic macromolecules, and that's really what Lab 6 is going to help us kind of figure out in lab today as well. So that's, timed pretty well for us, actually, as it turns out. Chapter 4, we're finally taking a step up away from molecule and to the level of cell. Now, why I'm saying this is a substantial step up, this is the first level at which life exists, right? We talked about that in Chapter 1, that atoms are not alive and molecules are not alive, but cells are the smallest thing, basically the building blocks of life. So we're going to spend several weeks, essentially, going over the basics of how cells work. And in Chapter 4, we're just kind of getting a sneak peek into that with this phrase that should look familiar: structure and function. Those are the same 2 things we paid attention to for macromolecules in Chapter 3, but now we're applying that idea to the entire cell. We want to think about what parts are found in a cell, what they look like, where they are, and also what are those parts doing that allow the cell overall to function. Chapter 4 is, sort of a big chapter because cells are the basic building block of life. We want to make sure we get the basic stuff down about them. There's a lot of intro stuff in this chapter, background sort of material. A lot of it, in my opinion, I'm going to say is almost like throwaway material because it's stuff you've either seen before in the lab book or you just know already from life. So I put a few slides in here where I'm going to gloss over the first several slides and you essentially won't have to worry about them. I'll, of course, make it clear which ones those are. We're going to start really then by taking a very quick look at prokaryotic cells. Now, you guys already know what that definition is. We had it in Chapter 1. Remember, that means cells with no nucleus. Remember, all cells have DNA because it's the genetic material for all organisms. And this distinction is whether it is or whether it's not found in that special compartment called the nucleus. This section on prokaryotic cells is really kind of a minority of this chapter. It's just a small part. We're going to spend much more of our time. The main focus, honestly, in this chapter is going to be cells like ours that have a whole bunch of special little compartments that are doing special jobs for the cell. And that will be, again, most of our time talking about what all these parts are doing and how that function contributes to the function of the cell overall. This first slide is one of those that I described as being sort of a throwaway slide, in my opinion. You guys already know that things look different with your naked eye versus when we look at them through the microscope. And because cells are so small, you can't really see cells that make up living things until you zoom in the way that we're, getting comfortable with doing in lab. The next 2 slides I'm going to have you cross off essentially. Sorry, I jumped ahead a bit. You can sort of cross this one off if you want to, and I'm only saying that because I'm not going to go over it in a detailed way. The cell theory is something you've seen before, or you should have in the intro to lab number 5 in your lab book. Cell theory essentially just states that all living things are made of cells, and cells can only come from other cells. They don't just like magically appear out of thin air. So this is an important part of biology, obviously, but not one that I'm going to spend a whole lot of time on, because I know you're seeing it in multiple places. And we established really in chapter 1 that all living things are made of cells. So I feel comfortable sort of glossing over this one. I'm going to say go ahead and cross off this figure, another throwaway figure in my opinion. I think everyone already knows that living things can be very little or very big, and that about covers it for that one. These 2 slides are the ones that I'm going to say cross off. Cell size. This information is something we're going to talk about next week in lab. So I feel totally fine telling you to skip it for right now. I'd rather wait and talk about it when it's more relevant to what we're going to do in lab, which is next week. So in terms of when you're sitting down to prepare for Wednesday's exam, ignore this slide and ignore the slide with the pictures of the cubes that goes along with it. I'll show you guys those exact slides next week in lab on Monday when we're starting lab 7, because there's a part of lab 7 that kind of goes exactly with these 2 slides. Okay, so that'll come back later, but in terms of for Wednesday, you're not worried about it. And that means we're sort of really beginning chapter 4, excuse me, with a reminder of a couple definitions you already know from chapter 1. I put them here in writing just to really make sure everyone remembers what these terms mean. This is sort of the first question you ask yourself about a cell when you're trying to figure out what it is that you're dealing with. The basic question is just, does this cell have a nucleus? Remember, for those of us that might not be as familiar yet, a nucleus is essentially a special little compartment within the cell that if the cell has one, that's where it puts its DNA. I always kind of talk about it like it's a little lockbox for safekeeping. Our cells are eukaryotic, along with the cells of everybody else in domain eukarya with us. Animals, plants, fungi, protists like amoeba. That's the big thing we all have in common. And we're going to come back to our cells essentially in a little bit. We're going to spend a little bit of time going over cells that are a little different from ours. In that one basic way, they don't have a nucleus. Remember, those cells still have DNA. Picture it just kind of floating around in the cell with all the other stuff that's there. It's not separated into its own distinct compartment. We are going to start with prokaryotes, and I want to sort of establish right away that in my mind this chapter does a lot with looking at differences. How are prokaryotes different from eukaryotes? Or once we're in this section of the chapter, how are animal cells different from plant cells? Because they're trying to make sure we recognize these major ways that different kinds of cells are different. But what I think is also equally important to do is establish a common thread, right? Similar to what we were doing in chapter 1, where we acknowledge like, hey, everything's really different, but there are a core set of features that every organism has in common. Likewise, there are a handful of parts that every kind of cell, bacteria, us, everybody has in common. And I want to make sure we don't lose sight of those similarities even as we're focused on differences. So what I'm going to have you guys do is I'll essentially say like, hey, somewhere off in the margin on the side of your paper, start crafting this list of parts that every cell has. And then we'll just put a few things on that list as we work our way through the chapter, just to make sure we don't miss sight of the fact or lose sight of the fact that certain parts are found in every kind of cell, regardless of what kind of cell it is. Okay, we're going to start, as I've already said a few times, with prokaryotes. And the real reason we're starting with these is that structurally, just parts-wise, they're a little bit less complex than our own cells. It doesn't mean they're simple, it doesn't mean they're easy. They're still very complicated, and in some ways more complicated than our cells. But parts-wise, there's less to tackle here. That's why we're starting with them. These kinds of cells also tend to be smaller than our cells. These sizes are not anything I would ever ask you. I only wrote them here to give you like a feel for it, because, I guess specifically because we've seen this unit in lab. Remember that prefix micro means 10 to the minus 6 or 1 millionth. So take a meter stick that we had in lab, chop it up into 1 million tiny pieces, and that's about the average size of a bacterial cell. The other way you have of relating, hopefully, to how small they are is think back to lab 2, that diversity of life lab where we had 1 billion samples kind of scattered all over the room. For most of the bacterial samples you saw, they were magnified 1,000 times, and they still looked pretty little, right? So that's just kind of my way of helping you relate in a few different ways to the fact that bacteria are generally very, very small. Remember I said in chapter 1, and I'm going to hold true to this, we're really not going to talk about archaea. Here in the book, they're clustered with bacteria simply because they also don't have a nucleus, but we're not going to talk at all about them. And this is really just a throwback to chapter 1. What I want to do is, again, talk about some of the basic parts that bacterial cells have. And we're going to do this with a few different slides. We're going to start off by talking about 2 to 3 layers kind of surrounding the outside of the cell before we turn our attention to what's on the inside. Appendages is a slide we're going to skip. I'll show you which one it is in just a minute. Okay. Before we start looking at the layers that surround the cell, I thought it might be nice to show you guys this image from the textbook because it lines up really nicely with some of the information that you had in your lab book in that same lab exercise 2, diversity of life. If you think back, and this feels like a super long time ago now to me, I don't know. When you saw those bacterial samples in lab, we had them labeled by what shape they were, and these definitions were in the lab intro. So I just thought, always it's nice when you're hearing something in lecture and in lab, I try to point out, hey, you've seen this in both places. So you saw a sample of bacteria that were round. That's what the term coccus means, or cocci is the plural, C-O-C-C-I at the end. Excuse me. You also saw some that were rod-shaped, or a bacillus. Bacilli, again just with an I at the end, is plural. And you saw a sporillum. Notice there's another kind of squiggly shaped one called a spirochete. You guys didn't see that one. And in all honesty, it would be sometimes hard to distinguish those two from one another because really what makes them different is sort of how flexible or rigid the shapes are. And it's tough to get a feel for that when you're just looking at like a slide with dead bacteria on it. So I would never ask you spirochete. Again, the real reason I'm showing you this slide is just to reinforce the shapes that you saw in lab a few weeks ago. One of the things I do think is interesting though that we can make a connection here is how are those different shapes kind of held in the proper shape, right? How is it that certain cells can maintain a very specific size and shape when they need to? And why I'm raising that question is because one of the parts we're going to hear about is exactly how the cells are able to do that, okay? So the term at the very top, cell envelope, that refers to kind of collectively whatever layers, plural layers, are surrounding the entire cell. And for bacteria, there are always at least 2, okay? So we're going to start closest to the inside and work our way out. The layer that you're always going to find, and this is true for bacteria, but also true for all cells. So here comes the first part you're adding to that little list in the margin I talked about, parts that every cell has, cell membrane. Every cell, yes, all bacteria, but also yes, animal, plant, fungi, protists, every kind of cell has a cell or a plasma membrane. Those two terms mean the same thing, so pick your favorite, whichever, okay? Excuse me. What I'm hoping we recognize, this is kind of a nice quick way to review some stuff from chapter 3, are these phospholipids, right? This was one of the types of lipids we spent some time looking at in chapter 3. And again, since your test is on Wednesday, let's quickly review. The circle portion of each phospholipid is called the phosphate head group. Remember, phosphate groups are polar. This is usually called the polar head group. Remember, polar, hydrophilic, dissolves in water or likes water. And the two tails you see, the non-polar, hydrophobic, fatty acid tails, they're meeting up in the middle to create this like water-free barrier layer, right? So inside the cell where there's a lot of water, we've got the polar head groups all pointed down. Outside the cell where there's also a lot of water, you have the polar head groups pointed up. and the parts that don't like water are all pointed towards each other in the middle. That structure that we saw in chapter 3, phospholipid bilayer. Now what you can see from this picture is there's some orange blobs there, right? So phospholipids aren't the only thing in the membrane, but they're the primary component. They're the major molecule that you're going to see in every kind of cell wall. We'll talk about what this other stuff is all about in chapter 5 that will start next week. So all bacteria, and also every kind of cell, has this cell membrane. You guys again know what this is doing. It's separating the inside of the cell from the outside. All bacteria, not all cells, but all bacteria, so don't add this to your list, have this additional layer outside the cell membrane called the cell wall. So if you're thinking bacteria, you're going to have cell membrane and then outside of it cell wall. This cell wall is more of a rigid structure, and it's not like it's, permanently stuck into place. It still has to have some give to it, but it has a lot more structure. It's not as fluid, if you will, as the cell membrane. And this is really the layer that answers that question I raised a moment ago. This is how cells, bless you, are maintaining their proper shape and size. It's because of how their peptidoglycan is organized in this cell wall layer. Now I'm hoping that somewhere in the back of your brain when I said and pointed to peptidoglycan, something lit up, because that's a molecule you've heard about before. As much as I can today, I'm going to reference previous chapters to make sure you guys haven't missed anything as you've been studying for Wednesday's exam. Peptidoglycan is a molecule we heard about in chapter 3. It's a polysaccharide, right, so big sugar. It's one of the structural polysaccharides we talked about. Remember, in general, carbohydrates can be used for one of two reasons, either for energy or for structure. For structure, we talked about 3 different ones. Cellulose, which is the cell wall for plants, chitin, which is the cell wall for fungi, and peptidoglycan, which is the cell wall for bacteria. So this is now the second chapter, second time you've heard about this molecule and what it's doing. Always, pick up on things like that. So for bacteria, you'll always have a cell membrane, and outside of that cell wall. Not all cells have a cell wall. For example, animal cells don't have that layer. That's why I specifically said don't put that on your list off to the side. We're going to skip this third layer, squiggle it out. Don't worry about glycocalyx. It's a third additional layer that some bacteria have. We're not going to go into it. So it's something we can let go and move on from. So it won't show up on the test. You don't need to worry about it. Once we move through the cell envelope, so for us, we heard cell membrane, cell wall. Once we move inside of those layers, we're inside the cell. We're in the cytoplasm. Cytoplasm is defined as being a semi-fluid solution within the plasma or cell membrane. I think about it like it's the goop inside the cell. It's the guts of the cell. Like once you're through the membrane, this is what you're in. What's in here? Well, obviously water and a bunch of other molecules, both organic ones. Remember, that means there are carbon and hydrogen there, as well as inorganic ones like maybe sodium chloride. Coming back to this idea of organic molecules, that's all of chapter 3. Right, so go back to chapter 3 and think about the kinds of molecules we talked about: carbohydrates and lipids and proteins and nucleic acids. All of those kinds of molecules, remember, why we spent a whole chapter talking about them is because they don't only show up in us and play those roles in us. Those kinds of molecules are found in all forms of life, including bacteria. So I listed a few sort of super important ones here I wanted to draw your attention to. Enzymes, which we'll spend a whole chapter on in chapter 6. This is a kind of protein. They speed up a chemical reaction. DNA, of course, I wanted to draw your attention to, and I also want to make sure you write this on your list of parts that every cell has. All organisms on the planet use DNA as their genetic material. So over on your list now you should have all cells have cell membrane, DNA. In all honesty, you could probably write just macromolecules on that list because all cells have carbohydrates, lipids, proteins, nucleic acids, but DNA is the big one I want to draw your attention to. Now, please be aware. Think about this carefully. All cells have DNA. It doesn't always have to be in a nucleus. Remember, bacteria do not have a nucleus, so their DNA, which they have, is kind of floating around in the cytoplasm with all the other stuff that's in there. Don't worry about the details of the structure of the DNA. I'm not going to ask you about chromosome or what it looks like or anything like that. I do want to point out this word simply because you might see this in your book or in your lab book, and I don't want it to confuse you. There is no nucleus in bacteria. That I know you know already. Nucleoid is just the term that people sometimes use to reference, like wherever the DNA is inside the bacterial cell, to reference like, oh, that's what I'm talking about. They just say it's in the nucleoid region. I'm not really ever going to use that term with you. I do not expect it to be on the test at all. I just wanted to point out what that was in case you see it again somewhere else. It's not the same thing as a nucleus. The other thing I want to mention here and also have you add to your list, ribosomes. The best way I think to describe what a ribosome is to call it a protein-making factory. Now you guys hopefully remember, again, just to reference chapter 3 again to make sure we're on it. Proteins were one of the hard categories, I think, to summarize function, because what I said instead was proteins have their hand involved in every aspect of cellular life, every structure, every process, every event you could possibly think of, proteins are involved with it. Well, all of those thousands of different proteins that we make to do all these really specific jobs, every single protein made in a cell is made by a ribosome. Ribosomes are critically important for the cell, and not just bacteria. This is, again, added to your list. So you now have like cell membrane, DNA, ribosomes. Every kind of cell must be able to make proteins. Every kind of cell does that because it has ribosomes. Okay. Any questions on these parts that you would find inside bacterial cells? Okay, here's that slide we're skipping. Cross it off, not coming back, not on the test. Don't worry about appendages. These are like some extra fun parts that bacteria can have that give them additional kind of functions. We're not going to go into it. This is a picture from the book to help you sort of visualize a bunch of the parts we've discussed. Now, obviously, a lot of the stuff up here we haven't talked about, or we skipped, or I said to cross off. So you need to make sure when you look at a picture like this, you look at it through the lens of like, what do I need to focus on? For example, we did not talk about, if you have slides, scribble all these off as I say them. We did not talk about flagellum, glycocalyx, conjugation pilus, fimbriae, inclusion body, or mesosome, right? So you have to remember that when you're looking at this sort of figure, because that really limits down what you need to see and what you should focus on, right? So let's go through it again. What did we talk about? So what can you look at this and kind of connect the dots with what I've discussed? This kind of thicker red layer right here, that's the cell wall. Remember, that's what's made of peptidoglycan, that structural polysaccharide we talked about in chapter 3. That's what allows these cells to maintain whatever shape they have. Whether they're a coccus or a bacillus or a sporulum, that layer is what keeps them in that correct shape. Moving underneath that, this thin blue layer, there's our cell membrane. That's our phospholipid bilayer. And again, we already kind of reviewed the structure of a phospholipid, so I won't say too much more about it. Once we're through that layer, now we're inside the cytoplasm. And there are really only two things that we focused on that are in there. This big scribbly line, that's the DNA, all the instructions for how to do everything inside that kind of cell. And then the other one are these little blue dots kind of scattered around everywhere. Those are the ribosomes, or our protein-making factories. Any questions on bacterial cells or the parts that we briefly went over? Okay.
So what did you say to cross out of?
All the ones we didn't go over. So flagella, glycocalyx, nucleoid you could cross off. I didn't say it the first time, but conjugation pilus, fimbriae, inclusion body, mesosome. So only, I really think that helps like remove visual clutter if you know which ones to focus on. Okay. The only thing you need from this slide is the very top bullet point. It's just a definition. And I'm only including it because it's something you saw in lab in exercise 2, that diversity of life one. You saw oscillatoria, which is an example of cyanobacterium. It's just a bacteria that can do photosynthesis. That's literally all you need from this slide. Nothing else. You ignore the rest. You don't even see it. You don't write it. You cross it off if you have it. All you need to know is that cyanobacteria are bacteria that can do photosynthesis. Not all of them can do it. That's why you give a special name to the ones who can. Any questions, bless you, on bacteria before we start talking about our own cells? Okay, like I said, that was a much smaller part of the chapter. We started there because structure-wise, there's not as many parts to talk about. When we start looking at our cells, you're going to notice pretty quickly there are a lot of specialized parts that our cells have that I didn't mention at all for bacteria. Again, structurally, eukaryotic cells are more complicated. The nucleus, in other words, isn't the only part we have that bacteria don't. And honestly, the rest of this chapter is going to be spent going over what are all these parts that our cells have, what are they doing, and how does that fit into the kind of big picture about how our cells work the way they do. I'm giving you, once again, sort of a big reminder here. You already know the definition of eukaryotic, cells that have a nucleus, and you already know that animals like us are not the only ones that this applies to. I think this is especially important to remember throughout chapter 4, because what you're going to see excuse me, is that in many of these cases, the book is going to continually show us a picture of an animal cell to kind of give us frame of reference, like, oh, here's the part we're talking about. And every time, almost every time, they show us an animal cell as a default. But I want you to really try to remember that all the parts we're hearing about are typically found in all of these cells as well. Okay, even though we're going to really sort of focus on us, what we're learning in this chapter applies across the board in many cases to all eukaryotic cells. And what we're really going to be focused on are these special little compartments called organelles. Many of them are going to be surrounded by membrane. The very same sort of phospholipid bilayer that we already saw makes up the cell membrane surrounding the whole entire cell. That's really what allows us to make these little compartments. It's like a little bubble of membrane, if you will, inside the cell that we can put certain things into. This is how we can start to get specialized function for all these little special compartments. Okay. Here comes another slide we're going to skip. We're going to not talk about endosymbiotic theory, which sort of kills me a little bit, but in the interest of time, I'm very focused on making sure we have enough time to get to what we need. So cross it off, not coming back, not on the test, which means I'm not going to show you the video that went with it either. And we're going to segue right into thinking more about these organelles. Okay, so make sure you make a note of the fact that we're skipping endosymbiotic theory. All right, so like I've already said a few times, organelles really are just little compartments that we have inside our cells. And because they are separated from each other and from the rest of the cytoplasm, these little compartments can become, and do become, very specialized. It's almost like a way for the cell to divide and conquer in a way. Like if we have all these different jobs that have to get done inside the cell for our cells to function efficiently and correctly, instead of trying to do all billion things at the same time in the same place and get confused and whatever, we're going to separate all these jobs and give one little job to each person or each organelle within the cell. That way, we know it's been handled and we cannot have to worry about or focus so much at every second about that one job getting done. We've delegated the task to someone who's capable of doing it. We are going to focus, or how this chapter is organized, in other words, is by dividing many of these specialized compartments into one of two sort of big groupings in the chapter. This first category is a big one. It's called the endomembrane system. Okay, always, I try to look for hints from a word in terms of what it might mean. Does anyone know what the prefix endo means? Into, or inside, something like that. So we're going to be seeing a bunch of membrane, phospholipid bilayer, But this membrane, like I referenced a moment ago, is going to be found inside the cell, kind of cordoning off all these different specialized compartments. Endomembrane system, complicated system of membrane found within the cell. And what these organelles that are going to be in this category all have in common, it's not simply that they're surrounded by membrane, because that's true of other organelles too that are not part of the system. What really binds them all together is that their function is part of the same overall pathway inside the cell. And the big goal in mind for this entire system is essentially to coordinate how to make and properly send to its destination some kind of product within the cell. Think about it like a little assembly line. We want to make something and send it to the next place to get tweaked, and send it to the next place to get tweaked, and send it then to wherever it needs to go once we've properly made whatever we're trying to make. And that's, again, what puts all of these many organelles we're going to see with each other in this endomembrane system. They all are involved in that little assembly line. They're all responsible for some aspect of it, and they have to communicate and coordinate to make sure that job gets done correctly. So we're going to see little tiny bubbles called vesicles. This really does just look like a little bubble surrounded by membrane. Why vesicles are so valuable is they can be used to efficiently move things around inside the cell. What do I mean by that? Well, I just kind of established this idea that we're going to be passing something, let's call it a protein to make it more concrete. We're going to follow how a protein is being made and then kind of sorted properly within the cell. Well, let's say I'm a ribosome, remember, protein making factory, and I make the protein. If I just spit it out when it's done, let's say the inside of the room is the inside of a cell. If I'm a ribosome up here and the protein I have just made is needed in the back corner of the room or the other side of the cell, if I just spit that protein out and hope it'll float over there eventually, maybe it would get there eventually, but it would not be a very efficient way to get things done, right? So we don't move things like that in the cell. Instead, what we do is, as I'm spitting out this protein, I package it into a vesicle. Think about it like a box car on a train car, right? Train cars can move all kinds of things, like cars or coal or whatever, right? I put it in a little train car and I send it on its way. Now, I could have said anything there, right? I could have said put it in a box, put it in a car. Why did I go for a train car? Think about how trains move. Can trains just go anywhere? No, right? They have to travel on railroad tracks. And I very specifically always use this analogy because that will allow us to understand how another system in the cell is working. There are railroad tracks made of proteins called the cytoskeleton network. So if I'm that ribosome, I'm going to package the protein into a little train car or a vesicle, drop it onto a railroad track, and send it exactly to where I need it to go. It's a much more efficient way to move things because it's directed. You're not just spinning it out and hoping it floats to the right spot. You're sending it exactly to where the cell needs it to go. And again, that's sort of a big process. You have to coordinate a lot of different steps to make something, tweak it the right way, and send it to its proper kind of location or destination inside or sometimes outside the cell. That's why we're going to see so many different organelles that are all part of this big system that have to cooperate and work together to make sure this process flows smoothly. Now, if it were me, I would put cytoskeleton right in this part of the chapter so we can see the clear connection. However, I always try to follow the book as closely as possible so that people who are going back and forth will have an easier time. For whatever reason in the book, sorry, they took cytoskeleton out and they put it in a separate section at the end of the chapter. So that's how I'm going to show it to you. We're going to go over all the parts of the endomembrane system, then we'll hit this second category, and then at the very end, we'll come back to the cytoskeleton network. I'll remind you of how it's linked to the endomembrane system. I just wanted to give you a heads up that it's going to be tackled in a different part of the chapter later. Before we tackle cytoskeleton network, we're going to talk about a couple, just two, organelles in the cell. They're both actually surrounded by membrane, but they're not part of the endomembrane system. They're doing very different jobs in the cell. And they're lumped in this little group just called energy-related organelles because mitochondria and chloroplasts are really important for metabolism in whatever kind of cell has them. And so we're going to get a brief reminder kind of back to chapter 1. What are these big important metabolic reactions I'm talking about? And what's happening really in each of these organelles. So we'll come back to that later today. The next 2 slides, excuse me, the next 2 slides have some nice pictures from the textbook. And I specifically include them in the PowerPoints, not because I'm going to stand up here and point to every part, specifically for people who might not have gotten a book. Now I know you have access to these figures because you can get them on Canvas. Right, so on this slide, you're seeing an animal cell. On the next slide, you're seeing the equivalent version, but of a plant cell. And what I want to do is just point out, these are here. These are, in my opinion, really good figures because they not only are labeling parts to show you where they are and what they look like, they're also capturing in a kind of quick little way the function of each part that's being labeled. So it's not so useful perhaps as an intro figure, like I put it here sort of in the beginning of the chapter. I think it's much more useful, both of these figures, as a review. Because once we've gone through and talked about all these different parts and what they do, you can come back here and sort of get a big recap all on one place. What I would rather use these figures for right now is to point out some of the differences that we're going to notice between animal cells and plant cells. And this this goes right along with part of Lab 5 that we were supposed to do last week. Part A of Lab 5 was geared specifically at looking at animal cells and plant cells. And there is a part of your lab book that asks you specifically to write down some of the major differences between the two. And so this is a really good place for me to take an opportunity and point those out to you. So most of those differences are things I'll have to flip to the next slide to show you, because a lot of the differences are things that plant cells have that our cells don't. But there's one thing here I want to point to that you're not going to find in a plant cell. There are these little 2 parts right here. There are always 2. They're always at right angles to each other. They're called centrioles. They kind of look like little pasta noodles to me. Okay, there's one centriole. There's another centriole. Notice there are 2 labels. Centrioles, 2 of them. The pair of them together makes up another part called the centrosome. So centriole, centriole, whole thing together, centrosome. That's one of the organelles we're going to talk about later when we go over the cytoskeleton network, those little railroad tracks I mentioned a moment ago. This is related to those railroad tracks. So when we talk about the railroad tracks, I'll double back and talk to you about what these do. But if you're building a little list of differences between animal cells and plant cells, number 1, animal cells have centrioles, plant cells don't. Okay. Also make sure you don't miss what makes this cell eukaryotic, the nucleus right in the middle. That's the part you're going to see on the plant cell picture on the next slide too, because of course, as you already know, plant cells are eukaryotic too. Now, there are 2 big differences I want to point out here. 2 parts these cells have that animal cells don't have, and a third sort of minor one I want to make you aware of as well. Big difference number 1, thick green layer surrounding the entire cell is the cell wall. We saw a cell wall earlier for bacteria, similar idea, just made of a different sugar, right? Remember, bacterial cell wall was made of peptidoglycan. Plant cell wall is made of cellulose. It's the one structural polysaccharide we actually looked at a diagram of in chapter 3 from the book, okay? Big difference. Plant cells have a cell wall, animal cells do not, okay? So that's our second difference. First difference was animal cells have centrioles. Second difference is plant cells have a cell wall. Third difference is another big one, this part right here called a chloroplast. Plant cells have them, animal cells don't. Chloroplasts are where photosynthesis happens, right? We as animals cannot do photosynthesis because we don't have this part in our cells. So it's a big difference, okay? Those are the big 3. Animal cells have centrioles, plant cells don't. whereas plant cells have a cell wall and chloroplasts, and animal cells don't. The sort of minor difference I always point out is this big bubble right in the middle called the central vacuole. You did not see that huge bubble on on animal cells on the last slide. Plant cells have a large central vacuole. Animal cells do not. Okay. That would be the fourth and more minor difference, I would say. As it turns out, some animal cells do have some vacuoles, but not this big one in the middle that serves the same purpose. So we'll talk more about that later. What I think it makes sense to start with is the part that defines these cells as being eukaryotic, which is, as you already know, the nucleus. Okay, so you already know some big important stuff about the nucleus. Number one, it's where the DNA is, right? That's like the big overall function of the nucleus. It contains all the DNA, that all important set of instructions for how to do everything. You already know it's surrounded by a membrane, right? A phospholipid bilayer separates inside of the nucleus from the cytoplasm and the rest of the cell. Here's a new part I haven't mentioned until now. That nuclear membrane, or nuclear envelope as it's sometimes called, has a bunch of little holes poked in it called nuclear pores. Those nuclear pores are really important because they leave a little tiny door open as a way for the rest of the cell to communicate with the nucleus. Those nuclear pores are tiny. Not everything can get through them. Our DNA, for example, our big bulky chromosomes, too big to fit through those pores. This is why the nucleus really does act like a lockbox. The DNA is trapped in there, but there are other things that have to be able to come in or go out. As an example of what are some of the things that might come in or go out, remember the ribosomes we talked about? Those little like protein making factories? Part of them are actually made inside the nucleus because, as it turns out, we have to access the DNA to know how to make them the right way. So part of them are made in a region that shows up darker inside the nucleus. It's called the nucleolus. It's a dark spot inside the nucleus where part of the ribosome is being made. We'll learn in just a second that ribosomes, fully formed functional ribosomes, are not found in the nucleus. So once these little parts of them are made, we ship them out through the holes to the cytoplasm, and that's where we finish building them and where they become functional. So I just think this is a nice example. While I'm talking about the nucleolus and what it is, it's a nice example of something that has to be able to get out of the nucleus to do its job somewhere else. I am going to tell you to cross the next slide off, okay? Structure of DNA, chromatin versus chromosomes, cross it off, not coming back, not on the test. We're going to tackle this information in a later chapter anyways, so we'll go into more detail about it there. And that means we're going to jump right to the picture. Okay, for most of the parts that we're going to talk about in this chapter, you're going to see a slide similar to this one, where they're basically showing you the little offset animal cell as a default, but remember, this could also be a plant cell or a fungal cell or an amoeba, because the part that we're talking about here, the nucleus, clearly is found in all eukaryotic cells. They're showing you the host cell for frame of reference, and then they have like a pop out in a brighter color of the part we're focused on. So the nucleus is generally shown kind of as a circle or an oval right near the middle of the cell. And they've pulled it out here for us. This darker spot in the middle is that nucleolus, where part of the ribosome is being made. That part, again, would be a good example of what would then pass through one of these nuclear pores to exit the nucleus. And I like that they're showing us a zoom in here to kind of reiterate the fact that this membrane is also a phospholipid bilayer, just like the cell membrane we looked at before. Any questions about the parts of a nucleus? Okay. We're going to cross off the next slide as well. This is, again, more detailed information we're really going to need to use later. Cross it off, not coming back, not on the test, pretend it was never here. Excuse me. And we will talk next briefly about ribosomes. Now, quite honestly, you already know the top bullet point, which is the function of ribosomes, why they are so important remember what I said earlier, they're little protein making factories. And you should already have them on your list of parts that every cell has. We already talked about these when we went over bacteria, because bacteria have them just like we do. And that's, I wrote a little reminder here, just in case you missed it when I said it a few times earlier. So most of this you already knew, okay? So let's focus on the new stuff. Where are these? Remember, structure and function. We know what they do. Where are they? What do they look like? In our cells, we have two options for where these ribosomes are going to be. Number one, like they were in bacteria, they can just be floating around in the cytoplasm. Those are called free-floating or just free ribosomes. You can cross off the parentheses here. Don't worry about polyribosome. We'll maybe talk about that later this semester. So that's option number one, floating around in the cytoplasm. Option number two, in our cells, not in bacterial cells, in our cells, they can actually become attached to another organelle, which is called the endoplasmic reticulum, or the ER for short. Okay. We actually have two kinds of ER in our cells, which we'll talk about in just a second, but the kind that has ribosomes attached to it, the ribosomes look like little bumps on the surface of the ER, so that kind, or that flavor, if you will, of ER that has ribosomes is called the rough ER, because the ribosomes look like little bumps, make it look rough. So again, in our cells, where a ribosome is, two choices, either floating around or attached to the rough ER. There is a figure on the next slide that, quite honestly, I only put here to remind myself to acknowledge to you that it's in the book, and to specifically say, ignore this. I used to not even put it in here, and then I would get a whole bunch of frantic questions over email when people were studying for the test, like, oh my gosh, this big figure looks really important. Like, no, cross this off. We're not coming back to it. I don't know why this is even in chapter 4. It's way too advanced for chapter 4. Cross it off, not coming back, don't have to worry about it, okay? This is essentially getting at why some ribosomes are attached and some aren't. We don't need to worry about that right now. As soon as I have started talking about the ER, we're now officially into that big assembly line issue I talked about earlier, the endomembrane system. So remember, we're going to see multiple components of the cell, multiple organelles, really kind of coordinating and communicating to work together to accomplish this big overall job in the cell of making and sort of correctly sorting a product to its destination. Even though I hadn't acknowledged it, we actually were already there when we were talking about the nucleus, because as I'll show you in a moment, the nuclear membrane or the nuclear envelope is actually continuous with the endoplasmic reticulum or the ER. It's all one connected layer of membrane. You can't really distinguish where one stops and the other begins. These are other parts we're going to talk about in a second as well. Okay, but first let's get into this really with the ER that I already made a mention of a few minutes ago. So the ER is, as it says in the top bullet point, really a complicated system of membrane. So what does that mean? Well, you know what a membrane is. It's a phospholipid bilayer, so we're going to see a whole bunch of membrane, but it is going to be much more complicated than the simple kind of boundary of the cell or of the nucleus that you've been thinking about it so far. We're going to see layers upon layers of membrane sort of folded back on itself, made into these tube shapes. Like, it'll just look much more interesting than being a simple line that separates in from out. This first type is a type I had mentioned already, rough ER. What makes this rough? Well, the presence of ribosomes studding the surface of the rough ER. Now, I'm hoping that this won't be too much of a leap for us. Since we've already gone over the fact that ribosomes are little protein-making factories, the presence of those ribosomes on the rough ER should lead you to this conclusion that the rough ER is probably producing proteins because of the ribosomes that are there. I'm going to say cross off glycoprotein. Don't worry about that. We'll get into that actually in chapter 5 a little bit. And this is just a nice little reminder from chapter 3. Proteins, while they're being made, will then assume their native conformation. You guys remember what native conformation is? or the whole idea of a pipe cleaner that you make into like a 3D sculpture. So as the ribosomes are spitting out these proteins, which you should know from chapter 3, is just a whole bunch of amino acids all strung together in whatever order. As they're being spit out, they're also folding into their correct 3D shape so that they'll be ready to go as soon as they're done being made. Now the other variety of ER that we have in the cell does not have ribosomes. No bumps, so it's called the smooth ER. Let's just fashion a guess. If there are no ribosomes, do you think proteins are being produced there? No. So what's going on instead? Production of lipids, like triglycerides, the fats and oils we talked about in chapter 3, like phospholipids. Don't worry about detox reactions. We're not going to talk about that at all. We're going to focus only on one thing for the smooth ER, and that is the production of lipids. So essentially, in the ER, we're going to see stuff being made. What stuff? Well, it depends. If you're in the rough ER, the stuff is proteins. If you're in the smooth ER, the stuff is lipids. So in the ER, whichever type, we're making something. We're either making a protein or we're making a lipid of some kind. Our next step, of course, is going to be to follow that protein and lipid to see where it's going. But before we start adding on, let's just kind of touch back to our picture so we can continue to build this picture of a cell we've been working on. Once again, as usual, they default us to an animal cell. But remember that big point I made right away? It could have just as easily here been a plant cell, a fungus cell, an amoeba. Anything eukaryotic could have been there. Here's that nuclear membrane with the pores that we talked about just a moment ago. And here's that point about the nuclear membrane really being continuous with the ER. It's not like there's a gap between them that, oh, that's the nuclear membrane and now this is ER. They really just kind of run into each other. These little yellow dots, as is labeled, represent the ribosomes. Remember our little protein-making factories? Present on the rough ER, absent from the smooth ER. Even if you ignore those little dots, the two structures still actually look a little bit different. The ribosomes are, of course, the main distinguishing difference, but notice that the rough ER kind of looks more squashed, and the smooth ER looks like tubes kind of coming out of the screen towards you. So they still do look a little bit different, but again, the big defining important difference is the presence or absence of ribosomes. Any questions about the ER, rough or smooth? Okay, why don't we go ahead and pause for a break? I'm going to cut our break a little bit shorter than I usually would because we still have quite a bit to get through. Let's do a 5-minute break today instead of 10. So be back at 11 and we'll finish off chapter 4. Okay guys, I know that was very quick, but we're going to go ahead and pick it up right where we left off. So we left off just before break by talking about the endoplasmic reticulum, where we're either going to make some kind of protein or some kind of lipid, depending on whether we're in the rough ER or the smooth ER respectively. So the next part I kind of mentioned already. You'll see it indicated on the slide in the next picture, but just to kind of remind you what the idea is here. Remember earlier when I said if we make something, we don't just want to spit it out and hope it floats to wherever it needs to go. As we're letting it go, as we're releasing it from the ER, we immediately package it into one of those vesicles, that little bubble of membrane, and then we send it to the next step. The next place we're going is an organelle called the Golgi apparatus, or the Golgi body, or the Golgi complex. I've seen all those different names used. As long as it says Golgi, it's the same thing. The Golgi has kind of this funny little nickname, stack of pancakes, which is actually quite fitting when you see what it looks like inside the cell. It's just several kind of stacks of flattened membrane. So a bunch of them just stacked on top of each other. Major functions, 2 of them. I wish I had written this slide differently, so I'm going to talk about it in such a way that I hope to highlight both of the major functions excuse me, that are going on in the ER. Sorry, the Golgi. Number 1 is the one I have written here. Modification of proteins and lipids. What does that mean? Well, remember, what we're bringing to the Golgi is some kind of product, either a protein or lipid we made in the ER, and we're essentially going to dump it in on one side of the Golgi, And as that protein or lipid is moving through that stack of pancakes, we're just going to tweak it a little bit. Maybe add a functional group over here, throw in a sugar molecule over there. We're just making some, however minor, adjustment to the product that we just made so that it's really ready to go do whatever job it needs to do in the cell. The second major thing the Golgi is doing, kind of like down here, move to its proper location. Put a number 2 there so you remember that's the second major function. As the Golgi is releasing this product from the other side, right? It came in on the bottom of the stack maybe, and it moves through and is going to get released from the top of the stack. As it gets released, the second big job of the Golgi is to make sure we're routing it to its proper destination. I always think about like the post office, right? You send a letter there, and then depending what instructions are on the letter, they send it to Alaska or to Maine or to Nevada or to wherever. They make sure it gets sent to the proper place it needs to go. That's the second big job of the Golgi. And that can be kind of one of 2 things. It can send it somewhere inside the cell. Maybe it's a protein that needs to be inside the nucleus or inside the chloroplast. It routes it to wherever it needs to go. But sometimes a certain protein or lipid is being made to go do its job actually outside of the cell that made it. And the Golgi recognizes that and flags it in a special way to get sent to the surface of the cell to be released. So again, 2 big jobs in the Golgi. Number 1, modification of whatever protein or lipid we made in the ER. And number 2, make sure we send it to the proper place when we let go of it on the other side. So what you're going to see on this diagram is a continuation of the picture we've been building, right? So here's the nucleus, nuclear membrane, rough ER, smooth ER. Here's our stack of pancakes, our new part, the Golgi. And what I want to point out is this little vesicle that I've been talking about. It's the first time we're actually seeing it, right? Vesicle is just a little bubble surrounded by membrane. And this specific vesicle, because there are several kinds we're going to see, this specific vesicle is named for what it's doing. It's simply being used to transport this green squiggle from the ER to the Golgi, so we call it a transport vesicle. That's it. So it kind of blebs off the ER, merges into the Golgi, and that green squiggle is simply passed from pancake to pancake, if you will, and then it gets spit out the other side. Now here's where I take just a little bit of an issue with this figure. I wish they had labeled it differently in the part that I'm going to show you next. They labeled this vesicle coming out the other side also as a transport vesicle, and that would be correct if that vesicle were going somewhere inside the cell, like back to the nucleus or over to the mitochondrion or whatever. But in this case, it's not really labeled correctly because look at what this vesicle is doing. It's going to the outside of the cell. It's being secreted. And when a vesicle is routed for the outside of the cell, it's given a different name, and one that's going to show up for us in a couple of minutes anyways. I wish they would just call it what it is from the beginning so you're not like learning different terms for the same thing. If you want to fix this figure kind of preemptively, this kind of vesicle is called a secretory vesicle, like secretary, but with an O, secretory, because it's going to be secreted away from the cell. Okay. So again, what is the Golgi doing? 2 big jobs. We're going to modify, which we can't really see in this figure. Again, add a sugar, add a functional group, whatever it is. We're going to modify the green squiggle, and then we sort it properly to its destination once we release it. I always ask a question of you guys on this slide, and that is to have you think about this green squiggle that I was talking about. What kind of molecule do you think that green squiggle is? A lipid. Good. 99.9% of the time people say protein, which is the wrong answer. Lipid is correct. Okay? If you didn't guess lipid, how the heck would you know that's a lipid? Only because it's colored in orange and you can see where it came from. That's the smooth ER. Smooth ER makes lipids. If that transport vesicle had been down here and outlined in purple, then it would have likely been a protein, right? You only know what that is because you can see where it is coming from. If I just showed you a random circle over here that wasn't either orange or purple, you would have no way of knowing what's inside of it. In other words, vesicles kind of all look the same from the outside. It's only if you know something more about it, like what's in it or where it came from, that you can make a better guess about what it probably has going on. I want to talk to you next about another kind of vesicle. So far you've heard about two on this slide: transport vesicle and secretory vesicle. The third kind of vesicle we're going to hear about looks the same, little bubble of membrane, but does a very different job, a very specialized job. And the name of this vesicle is a lysosome. Lysosomes play a critically important role in the cell. They are considered to be part of the endomembrane system because they are produced by the Golgi. And what I want to talk to you about first, I think, is what's inside of them. Remember, all vesicles look the same. It's really what's inside of them that tell you what's going on. And there are two big important features of lysosomes that I want to highlight for you. Number one, very low pH. So we talked about the pH scale in chapter 2, which feels like a lifetime ago somehow. If we're talking a low number, like 2 or 3 or 4, is that acidic or is that basic? Acidic, good. Low numbers are acidic. So it's very acidic inside the lysosome. Important property number 2, lysosomes contain powerful hydrolytic enzymes. Let's kind of pull that apart a little bit. Enzymes are just proteins that speed up some kind of reaction. I did not use the word hydrolytic, but we have talked about hydrolysis reactions. Do you guys remember what hydrolysis reactions are? This is a big review from chapter 3. Right, you add a water and you break something down into smaller pieces. Think about it like chop, 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 chop down into little pieces. So when we kind of marry these two things together, inside lysosomes, you're going to find powerful enzymes that chop stuff up into smaller pieces. Along with the acid. Why am I mentioning the acid again? Again, to review from chapter 3. I want you to think about how we talked so much about proteins folding into the right shape. That native conformation I already reviewed kind of briefly earlier. Remember, that shape in large part is held together by hydrogen bonds, the weakest kind of bond we learned about. And one of the things I told you guys about that structure is that even once we've kind of put it in place, it's not permanently there. That structure can be disrupted, or proteins can be denatured by a change in temperature or a change in pH. Most proteins, when you throw them into a very acidic environment, they just unfold, right? I take your 3D pipe cleaner sculpture and I just pull the ends of it straight. It unfolds it, it opens it up. And then chop, 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 chop. Lysosomes are like the garbage disposal inside the cell. We send stuff in there to be broken down into smaller pieces, to be destroyed essentially. This is very useful in two different kind of scenarios for the cell. Number one, if there's something coming into the cell and we might not know what it is, we send it to the lysosome. It's like a first line of self-defense. What if it's a virus or a bacterium that might be trying to make us sick? We send it to the lysosome and we try to break it up and destroy it before it could make us sick. Or number two, if there's like an old part of the cell sitting around that's not doing its job properly anymore, send it in the lysosome to make sure we break it down and can then recycle the pieces, right? We're not like making them all disappear, we're chopping them into smaller pieces, and then we can use those pieces to build something new and useful for the cell. So again, I think about lysosomes like they're the garbage disposal. There's not really, in all honesty, a great picture to show you for this, because from the outside, all vesicles kind of look the same, right? So if I lined up a transport vesicle, a secretory vesicle, and a lysosome, and didn't tell you anything about what was inside of them, you would not be able to tell them apart, right? And that's why, this picture, it's not all that useful, because there's just a little circle that they've highlighted for you. And unless this picture were labeled as it is, we wouldn't know what's going on here. What they're showing us in this top picture, what I like about this is you can see the phospholipid membrane bilayer surrounding the whole lysosome, but these are just a couple like old parts of the cell inside of it. Usually, I try to get at how important lysosomes are by talking about what can happen if they don't work properly. I'm going to skip that for us because we're short on time. Oh, I hate skipping this. Cross off this bottom part. We're not going to talk about Tay-Sachs disease, which is a fatal genetic disorder, which is pretty severe. If you're curious, you can ask me. I'll tell you about it later. I just don't want to take the time in lecture to go over it. So skip that. Not coming back, not on the test. Okay. And my computer is about to die, which is just perfect. Okay. I really like this figure a lot because what this can give us is a beautiful way to review all of the parts of the endomembrane system that we've talked about so far. I'm just getting Canvas opened up so I can jump over to the slides on here if I need to. The first thing I want you to notice is that there are two different labels at the top of this slide. Picture it as though there were like an imaginary dotted line drawn right down the middle. The endomembrane system is involved in two separate and important processes in the cell. And so I want to go over both of them with you. I'm going to start on the right-hand side with what the book had labeled as secretion, making and secreting or sending away some kind of product. Notice I added an extra label for this. Again, like I said earlier, I'm a big fan of call it what it is from the very beginning. This term, exocytosis, is one that you're going to hear in chapter 5 anyways to describe this exact same process. So I wish they would have just put it in here in chapter 4. Cyto means cell. Any guess as to what exo means? Out. Perfect. So exocytosis is the process by which we move something out of the cell. And this is what we already saw happening in that figure earlier. Okay, so let's kind of track progress from right where we started, half an hour ago or whatever. We've got our nuclear membrane, the rough ER with the ribosomes, and now you can see the blue squiggle is labeled protein that's packaged in that transport vesicle. If that transport vesicle is coming from the smooth ER with no ribosomes, here's a label to confirm what you guys said earlier. It's likely a lipid coming from the smooth ER. Regardless of whether it's protein coming from rough or a lipid coming from smooth, we package it into a transport vesicle, a little boxcar, right? Sends it on those railroad tracks to the Golgi. We dump it into a little stack of pancakes here, and remember what's going on there. Some kind of modification is likely occurring. Add a functional group, tack on a sugar, cut off something, whatever it might be. We're making an adjustment to the lipid or the protein that we just made. After we're done modifying, we're going to dump it out the other end of the Golgi. And here's that term I told you about before, secretory vesicle. In this case, because we're seeing this blue product being secreted from the cell, we can label that vesicle properly as a secretory vesicle. Now, whenever you have two things surrounded by membrane kind of come together, picture it like you're blowing bubbles, right? If you have two little bubbles that come together, they just merge together. So this secretory vesicle approaches the membrane, and it just kind of becomes part of the membrane, and the contents get dumped to the outside. That is the process of exocytosis. Any questions on how we saw kind of coordinated communication between all these various parts in the endomembrane system to allow that process to happen? Okay, so now we're going to turn our attention to the left-hand side of the screen, looking at this process called endocytosis. We saw the prefix endo already today. You guys know it means in. Cyto still means cell. Endocytosis is the process by which we bring something into the cell. And the reason why this is part of the endomembrane system is because of the role of these lysosomes. Remember, lysosomes are made by the Golgi. So for example, maybe the enzymes that are in there were proteins that had been made by the rough, got trafficked through here, a bunch of acid got dumped in, and those functional enzymes are packaged in with all the acid. That's what makes that vesicle a lysosome. So the new bit we're seeing is that something, some orange blob, whatever it might be, virus, bacterial cell, giant macromolecule, who knows, it's a big question mark for us. Some big orange blob is coming in. We package it into a vesicle. There's what, the fourth kind of vesicle we've seen? Just incoming vesicle. We don't know what that is. it could potentially be something that would be harmful. We don't want to give it a chance to hurt us. So again, kind of first line of self-defense, you're going to see this fusion event between this incoming vesicle and the lysosome. That moment when they come together, remember, two little bubbles merge into one. I picture that like an unleashing of the hounds kind of an event. The acid causes that thing, whatever it is, to kind of open up and unfold, which makes it more accessible for all those hydrolytic enzymes to chop it up. And after we see that, excuse me, happening, we go from one big orange blob to smaller orange chunks, right? The lysosome has properly acted like the garbage disposal I called it earlier. Any questions about any part of the endomembrane system or how those parts function in both exocytosis and endocytosis? Okay, so we've seen at this point, what, 3 or 4 different kinds of vesicles that were all part of this system. Transport, secretory, lysosomes, incoming vesicles. I mentioned earlier, and I want to reiterate, that not every vesicle is part of this system. Vesicle is almost like a generic term for any little bubble surrounded by membrane, and there are a couple of other kinds of vesicles that I want to talk to you about. Actually, just one more. We're going to limit this down. I have a slide here. I think it's next. We're going to cross it off. So we're not going to go over peroxisomes or what they do. I'm fine with that because we weren't really going to talk more about them later anyways. We're done with it. What I do want to talk to you about, though, are a type of vesicle called vacuoles. Okay, this is the only type of vesicle we're talking about, essentially, that's not part of the endomembrane system. So just remember it as kind of like the odd man out of that regard. So again, if you had slides printed, cross off everything about the peroxisome, not going over it, not coming back, not on the test. That gives us more time to talk about vacuoles. You guys know I like analogies. Okay, so how I think about vacuoles. Tupperware container. Tupperware container inside the cell. Why do I say that? Why do I think about it that way? What do you do with Tupperware? You just store stuff in it, right? That's really what vacuoles are doing for the cell. It's a little, or sometimes bigger, storage vesicle. Vesicle just surrounded by membrane. You just pump stuff in there and hold on to it for essentially as long as you want to. What kinds of stuff? Well, really anything. Water, you could put sugars there, you could put lipids there, you could put food molecules there, waste products, pigment molecules, like really sort of anything could be pumped into it. Don't worry about these specialized vacuoles. I really only list those because those parts show up in your lab book in lab exercise 5. We're not going to focus on them even in lab in a big way at all, so don't worry about that, okay? I did want to touch on this point simply because I mentioned it earlier. Historically, it was thought that animal cells did not have vacuoles, but now we know there are a few exceptions. And this, I feel, always sounds like a trick question, but I promise you it is not. Good example of where we have these vacuoles in our cells is inside fat cells. So can you think of what substance we might be storing a ton of inside fat cells? Fat, absolutely, right? It sounds like it's a trick question. It's like, can it be that easy? Yeah, it's that easy. Fat, lipid molecules. If you were to look at a fat cell, also called an adipocyte, under a microscope, the nucleus, which we usually think of as like the biggest part inside a cell, is like smushed off to the side, and the vast majority of the inside of the cell is just a massive vacuole packed to the brim with lipid molecules. You're just storing a bunch of fat inside that vacuole, right? So there are exceptions. There are animal cells that do have vacuoles. We're going to focus more on the role that they play in plant cells. And specifically, we're going to focus on the one big one that all plant cells have, kind of right near the middle, that takes up a huge part of the inside of the cell. It's called the central vacuole. Finally, because they have to, finally we're seeing a plant cell for reference here, right? This is that giant, looks like a bubble almost in the middle of the cell I pointed out on those big overview figures, and it can take up a massive part of the interior of the cell. 75–90% of the inside of the cell can be central vacuole, okay? So what's in there? Well, the same kind of stuff I already had mentioned. water, sugar, salt, lipids, waste products, nutrient products, pigments, like really you could put anything in there. We typically call what's in there sap. And I'm mentioning this not only because it's a difference between animal cells and plant cells, right? Animal cells don't have what's called the central vacuole like plant cells do, but I'm also mentioning this because we're going to come back to it next week in chapter 5. When we talk about how plant cells can essentially respond to environmental conditions. If a plant cell finds itself in a situation where it needs to dump a bunch of water to quickly adjust, it has a reservoir to quickly take the water from, dump it all out. Or if the plant cell needs to take on a bunch of water to quickly adjust to its surroundings, bam, we have a huge holding tank to pump it straight into. The presence of that large central vacuole gives plant cells kind of a lot of flexibility in terms of how quickly they can react to changes in their environmental conditions. And that's something, again, we'll discuss in chapter 5. Any questions on this large central vacuole that plant cells have? Okay. Question. Is this part of the endomembrane system? No. Nope. No. Remember that was, see, I told you I was going to die. That was the one type of vesicle that we talked about that is not part of the endomembrane system. Let me open this up. Okay. That slide is sort of the end of that first big part of the chapter where we looked at the endomembrane system and other kinds of vesicles. What that means is we're ready to transition into the second kind of grouping, which is much smaller and faster, of organelles inside the cell, the energy-related organelles. And why I said it's a much smaller and faster grouping is this really only has 2 things in it, as opposed to the endomembrane system that had a whole bunch of different parts. The energy-related organelles, mitochondria, chloroplasts, just those 2, that's it. So I'm hoping we can sort of draw on some of the stuff we talked about in chapter 1 when we went over metabolism as like a requirement for life, right? The ability to get food and energy. We talked a little bit about energy and the fact that all living things have to get it somehow. This is right within that wheelhouse, okay? So both chloroplasts and mitochondria are the site of, are very important for some big, important metabolic reactions. So let's talk about chloroplasts first. The big thing happening inside chloroplasts is the process of photosynthesis. And again, in chapter 1, we went over the fact that photosynthesis is essentially just a big energy conversion. Plants and other photosynthesizers are essentially just capturing the energy in sunlight when it's available and using that energy to build a molecule of sugar. Right? This is a covalent bond with my fingers, to hook together 2 carbon atoms to build a sugar. So they take sunlight when it's there and they can store it in the form of a molecule, a physical object. And again, the point I always make is if it's midnight, how much sunlight is available? None, right? Because it's dark. So if you as a plant were dependent on sunlight, you'd be screwed at midnight unless you had been able to make a sugar. Now you've got the sugar just hanging out in your pocket. You pull it out. Now you've got energy to use at midnight even when it's dark. We're going to focus on photosynthesis and more about the structure of chloroplasts in chapter 7. That whole chapter is about photosynthesis. Okay. Mitochondria are also very important for metabolism, but they do sort of the opposite in many ways of what photosynthesis is. What's happening inside mitochondria is a process called cellular respiration. Cellular respiration is how we in our cells take a molecule of sugar, like the one that was built by plants during photosynthesis, and we are now going to rip it apart to release the energy that was stored there. We release the energy and we convert it into a form that we're able to use much more easily and quickly, something we talked about at the end of chapter 3, ATP. This is why mitochondria are always that way overused, like there are memes about it, the powerhouse of the cell, right? That's why mitochondria are called the powerhouse of the cell, because it is the site of the majority of ATP production. ATP is our favorite go-to form of energy, right? So within mitochondria, we take a sugar, we rip it apart, And as energy is released, we convert it to ATP so we can use it. Now, the big important point I want to make here about mitochondria and why I'm making such a big deal about this is because I see a lot of people mentally fall into this. Okay, plant cells have chloroplasts and animal cells have mitochondria. Like, all of that is correct, but you're missing a big piece of it when you do that. Found in, you can ignore the almost because we're never going to talk about any exceptions. Mitochondria are found in all eukaryotic cells. What does eukaryotic mean? Have a nucleus. Okay, let's get right to it. Do plant cells have a nucleus? Yes, they do. So do plant cells have mitochondria? Yes, they do. Right? A lot of people do like chloroplast, plant cell, mitochondria, animal cell. Plant cells have both chloroplasts and mitochondria. They make the sugar in the chloroplast, then at midnight they can use it in the mitochondria. So you have to remember that plant cells have both chloroplasts and mitochondria. Plants do both photosynthesis and cellular respiration. I'm clearly making such a big deal about this because I see a lot of people sort of forget that second half, that plants do both. We skipped endosymbiosis earlier, so we're not going back to it now. Cross off that last bullet point. And we'll take just a quick look at chloroplasts and mitochondria. Again, quick look. We're going to focus more on these structures in Chapter 7 and Chapter 8, respectively. This is just our first glance. Once again, because they have to, they're finally showing us a plant cell here. And what you're getting a view of is the fact that chloroplasts have a complicated structure. They also have membrane like all the other parts we saw earlier, but they don't just have a layer of membrane separating inside from out. They have an outer and inner membrane surrounding the whole thing, and they have a third layer of membrane, the more interesting one, called the thylakoid membrane, folded into all these little disc shapes that are stacked up. We're going to go over all those parts in detail in Chapter 7. So like for Wednesday's test, don't worry about all these terms, right? The more important connection to have in place is that plant cells have chloroplasts. Chloroplasts are used for photosynthesis. If you have that kind of streamlined down, you're good. Okay. Questions on chloroplasts? All right. Mitochondria. Forgot. Wrong computer. All right. Couple important points I've already made to remind you of, again, top bullet point, found in plants, animals, algae, fungus, protists. If, for this class, essentially, if it has a nucleus, it also has mitochondria, plural, right? Mitochondria is the plural version of that term, and that's the term you hear most often because cells tend to have hundreds or thousands of mitochondria per cell. It does vary a bit depending on cell type. Something like a fat cell that's just sitting there to store fat doesn't need as much, powerhouse ability as something like a muscle cell that's moving all the time and burning through lots of energy. So there are some differences, but in general, just assume there are a ton of mitochondria inside cells. I want to once again focus on that way overused but very useful nickname, powerhouse of the cell. This one's even better, I think, than stack of pancakes for the Golgi. Stack of pancakes tells us what it looks like. Powerhouse of the cell for the mitochondria tells us what it's doing. This is the primary site in our cells and plant cells and fungal cells and amoeba, etc., for ATP production. Remember, ATP is our second-to-second, minute-to-minute go-to form of energy, and this is where we make most of it. We're not going to talk about any mitochondrial diseases, so you can get rid of that. We're not coming back to it and not on the test. And once again, I'm just going to show you kind of quickly structure of mitochondria. And again, mitochondrion this time, that's what it is when it's just one. That's a singular version of the term. Much like the chloroplasts, we've got a couple membranes here, this time only two actually. The outer, which is wrapped around the whole thing, and the inner, which kind of folds into the middle. We'll go over those details in Chapter 8. Don't worry about them for now. This is where I specifically hate that they show you an animal cell because it buys into people's preconceived notion that these are found only in animal cells. This is where I especially wish they had put a plant cell and an animal cell to remind you of what I've already stressed. If it has a nucleus, it also has mitochondria. Okay. Should you put this on your list of parts that every cell has? No. Why? Do bacteria have a nucleus? No. So do bacteria have mitochondria? No, they do not. Okay, so make sure, I wanted to specify that. If it has a nucleus, it has mitochondria, but not everything has a nucleus. Any question on chloroplasts or mitochondria? Our two energy-related organelles. Okay, so we've got kind of one system, I guess, left to talk about at the end. It's the one I mentioned earlier as those like railroad tracks at the beginning of class, the cytoskeleton network. The cytoskeleton is made up of a bunch of proteins that are found all over the place inside the cell in these very organized structures that really do kind of look like little railroad tracks. We're going to start by tackling overall function. There are two. The cytoskeleton is very important for, number one, helping cells maintain their proper shape. And this is something that I especially highlight for animal cells. Remember how I stressed earlier, animal cells do not have a cell wall? Plant cells have a cell wall made of cellulose. Bacteria have a cell wall made of peptidoglycan. Fungi have a cell wall made of chitin. Those other kinds of cells have that like rigid surrounding layer that helps a cell be the right shape. Animal cells don't have that. So in order for our cells to be the right shape and size, we must have some other thing to rely on. And what we rely on for that function is our cytoskeleton network. Many of these proteins, as you'll see shortly, they kind of form like a scaffolding, if you will, that the cell membrane like rests upon. So by rearranging that protein scaffold where we want it, where we don't, that's how we can make sure the membrane of the cell is in the right place. The fact that we can rearrange it is something I'll come back to in just a second. I want to make sure I hit the second function first. It's the one I talked about already with that whole idea of the railroad tracks. Cytoskeleton is useful for movement in a couple different contexts, in a couple different ways. Number one, you can move something like a vesicle, which is how I talked about it earlier. You can move a vesicle around on a cytoskeleton, like railroad track. But also sometimes the entire cell itself can move by rearranging, again, moving, by rearranging these protein structures to wherever it wants to go. So the whole idea that this system is dynamic, again, opposite of static. Static would mean once you build it, it's there permanently forever, never moving. Dynamic means we can change this. Essentially, these proteins, the cell makes a whole bunch of this protein and can build it into these long chains. And let's say I've got a railroad track going out there and I realize that I'm not really using it anymore. That it's not serving a purpose then, right? So it's useless, it's wasteful. So instead of leaving it there forever, I then have the ability to break it down one piece at a time, maybe build a new railroad track going over here if I need one. The cell can constantly rearrange, so there's a railroad track going anywhere you might need one at any given moment when you need it. It gives the cell a lot of flexibility and control over both where the edge of the cell is and also where and how things are moving inside the cell. So this is a really important part of the system. There are multiple types of proteins found in the system. We're going to trim it just down to two. We're going to talk about actin and we're going to talk about microtubules. We are not going to talk about intermediate filaments at all. Okay, there's a slide here, but I'll tell you to cross it off when we get to it. We're going to do actin first simply because it's, the smaller of the two. They're smaller protein filaments. What I want to do first is make sure we know what we're looking at here. Each one of these green balls is a fully formed and folded protein. So to make one of these green circles, we had to put together a bunch of amino acids. It had to fold up into its 3D shape. Bam, now we've got one green circle. So we make a whole bunch of this actin protein. And once we have a bunch of the protein, then the proteins assemble into these little fibers, two little ropes kind of twisted together. This is what the cytoskeleton network looks like. A whole bunch of these protein filaments all over the place inside the cell. Now this figure is kind of complex in my opinion. I want to just briefly talk about what this is trying to show you. The actin itself is shown in green, and the other colors are other components of the cell that we're being asked to think about kind of how they interact with each other. In blue is the cell membrane, phospholipid bilayer, you guys know the deal with that. In pink is another protein, sometimes called a motor protein. And I want you to picture this like it's my arm. My elbow is attached to the membrane, like it's planted, it's not gonna move. But this part of my arm, I can grab the rope and move it and let go, right? So if this were actually a rope, this is the whole point, that I could like move the rope even though my elbow is planted. This is trying, I don't think it does a great job, but it's trying to make the point about how actin filaments can be used for movement. Oftentimes, it's movement of the cell itself, and I'll talk to you about an example of that a little bit later. But sometimes these are also used like railroad tracks, as I've been talking about. So making sure we connect with the function. Last slide kind of got a structure. Here we want to make sure we connect the dots to what the cytoskeleton network does. Actin fulfills both roles. Actin really does help cells maintain their shape because, as I mentioned earlier, those actin filaments, we saw one of them on the last slide, that one green rope thing, right? Those are found all over the place throughout the entire inside of the cell. It really does form this like dense complex network that goes right up to the edge of the cell. It really does form like a scaffolding that the cell membrane then kind of sits on and rests upon. That helps us establish where the boundary of the cell is. And actin is also important for movement. Sometimes of organelles, excuse me, although usually that's the other kind of cytoskeleton we're going to talk about in a minute, the microtubules. I think a lot of people are surprised by this, that chloroplasts actually can move around inside plant cells. Most of the time when we see them, it's like a drawing or a diagram, and it's just in one spot. Next week in lab, I think you'll get a chance to see. If you watch a chloroplast inside a cell, it actually looks like it's wiggling around or moving around the edges, and chloroplasts do actually move on actin filaments. This is the example I usually focus on, and this is why I'm going to talk about it later when we talk maybe on Wednesday about what Lab 5 was supposed to be for you. You would have been looking at live specimen of amoeba, like living organisms, and amoeba are capable of moving in a very specific way, and how they do that is based on them rearranging their actin. They build actin out in one direction and it forces their membrane to go out. At the same time, if they break it down or retract it, it'll bring up the rear. So the whole cell essentially crawls across the surface of the microscope slide simply based on how it's rearranging its actin. And I'm going to try to find you guys a video so you can see that even though you didn't get to see it in lab. Any questions on actin as an example of cytoskeleton protein? Okay. Again, cross off intermediate filaments, not coming back, not on the test. I'd rather spend our time talking about microtubules. Okay? Microtubules are also part of the cytoskeleton. They look a bit different than actin. They visually, I always think about like an empty roll of toilet paper. As weird as that sounds, think about the cardboard, right? It's a little cylinder, kind of short, hollow in the middle. The cardboard is the protein, it's called tubulin, that makes these structures up. I'm going to show you a picture on the next slide just to give you a visual. These details are not critically important. It's just to kind of connect the dots with the picture. What's more important that I always want to point out is that these are the railroad tracks that I always talk about. When I say railroad tracks, I really mean microtubules, because by and large, this is the cytoskeleton protein that's used for moving vesicles, which is the main way that we talked about this earlier. You guys already know that the system is dynamic, meaning we can break down and build new railroad tracks whenever we want to, wherever we want to. That, I think, raises the interesting question: how does the cell know what to do, where to build them, when to break them down. There has to be something in charge of making those decisions. And that's what this MTOC does. It stands for Microtubule Organizing Center. It's like the boss of all of the microtubules inside the cell. That MTOC is what dictates where the railroad tracks are going at any given moment in time. It's a really important structure. In our cells, the main boss here is the centrosome, which is that part that's made of the two centrioles I mentioned to you earlier. So we're going to come back to this in just a second. To me, it makes sense to look at microtubules first because the centrosome is actually made of microtubules. It's the boss of all of them, sure, but it's also made of microtubules. So you have to understand microtubules first before you can look at that. All right, so here's that visual empty toilet paper roll idea, right? The yellow and the orange are the actual proteins that these structures are made of. And once again, I just want to talk to you briefly. Details are not important here about how these are serving as railroad tracks. It once again would require the use of another protein called a motor protein that actually helps with movement. Here you're seeing a vesicle, any kind of vesicle we talked about, transport vesicle, secretory vesicle, you get the idea. The vesicle is attached to the motor protein on one side, and these motor proteins essentially change shape to look like they're walking right down the microtubules. And since the vesicle is attached, when the motor protein moves, it drags the vesicle along with it, right? Train car, railroad track. Some motor proteins walk forwards, other motor proteins walk backwards. Again, it gives the cell a lot of flexibility in terms of where we're moving things. Any questions on our railroad tracks, microtubules? Okay. For both actin and microtubules, there's one more view of them I want you to see in the cell, and that's why I'm showing you what looks like a big scary, image from the book. There are only two new parts of this you're going to see. First of all, we skipped intermediate filaments, so the whole middle part goes away. You already saw these pictures, so that's nothing new. We're going to ignore these guys over here. The two parts I want you to see are these two pictures. What those pictures represent are microscope images of actual cells where those proteins have been flagged with a fluorescent dye so you can see where they are. And why I like to show these diagrams or these images is because I really think, especially for actin, this is a visual confirmation of what I've said all along, that actin forms this dense, complex web that makes like a scaffolding that the membrane sort of rests upon, right? The membrane of this cell is not colored in any kind of way. But can you see the outline of that cell exactly where it is? Absolutely. Because where the actin stops, that's where the membrane would be sitting. And that's where the edge of the cell is, right? So again, especially for animal cells like ours, this system is what helps maintain the proper shape of the cell. Down here, what I like for the microtubules is that showing you a visual on where all those railroad tracks are going. There are a ton of them, again, going to every boundary of the cell. And remember, this is a dynamic system. So if we're not using this one right here, it would be broken down and rerouted to someplace more valuable, more useful. Any questions on cytoskeleton network? Okay. There are just a couple other parts I want to talk to you about. This one I've already mentioned a couple of times, so it's really just to kind of go back and loop it together, essentially. Centrioles, okay? First of all, I want to repeat what I said earlier. This right here is one centriole. This other postanoodle is a second centriole. One centriole, two centrioles. They're always found as a pair like that, and the pair of centrioles is called the centrosome. That's a term I just mentioned like a couple slides ago. Remember what the centrosome is. MTOC, Microtubule Organizing Center. This structure is like the boss of all the microtubules, of all the railroad tracks found throughout the entire cell. That's why it's usually located right near the nucleus in the middle. It's a good spot to have a command center because it's a central point of command. Now, hopefully we can make a little connection here. What we talked about since the beginning of class today is that the railroad tracks are used to move things. That's what microtubules are useful for. That's what the centrioles are really controlling. And we're going to hear much more about the important role of these at a specific time for every cell during cell division. Like if you've ever learned mitosis or meiosis before, the centrosome, the centrioles, play a key role in moving around some really important stuff during cell division, the chromosomes that are being divided between daughter cells. So we're going to kind of repeat this. We'll come back to this in chapters 9 and 10 later this semester. These structures, the microtubules that form the basis for the centrioles and the centrosome, they're also used in two other structures. Just going to talk briefly about them only because they were in lab as well, okay? Two structures used for movement, cilia and flagella, okay? This is essentially the last slide. There's like a summary table, and then there's the recap of the whole chapter. There's a crazy picture slide next, which is just going to be a skip it. This is where you're getting what I want you to get. Cilia and flagella are sometimes found on cells, and when they are, they're used for movement. We're not going to go into details of how these are like moving. It's related to microtubules that are found like at the base of their structure. What's the difference here? Cilia are a lot shorter and a lot thinner. They're like tiny little hairs, if you will, covering the entire surface of the cell. And you are going to see these in the handout I put together for you for lab exercise 5 that we missed last week, because one of the organisms you're going to look at, Paramecium, is covered with cilia. And as those little hairs move, the cell itself is like swimming around. It propels movement of the entire cell. Flagellum. Flagella is plural, flagellum is singular. It's also used for movement, but it's a much different looking structure. It's much bigger and longer. It looks like a tail or a whip coming out of one end of the cell. If you were someone who saw a sperm cell when we were first using our microscopes, that's the kind of mental default that a lot of people think of for a flagellum from people. If you look at a sperm cell over there, the long tail that you usually would think of on a sperm cell, that's a giant flagellum, right? So again, it powers movement of the cell. All that I ask you know for this part, this slide goes away. It's a crazy picture from the book, way too complex, I think, at this point, is that cilia and flagella are used for movement. And I always talk about it at least briefly because it ties into lab 5. What we're going to end with, almost perfectly timed. I wish I was a minute quicker, I suppose, but I just wanted you to be aware that there's a summary table in here. This is going to give you a yes or a no for all the different parts we talked about for the 3 main kinds of cells we went over. For prokaryotic cells, excuse me, notice there are a whole lot of no's, no, 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 because those cells, again, structurally are more simple. Over for the animal and plant cells, you're going to see some of the big differences we talked about are captured there. What do animal cells have that plant cells don't, and vice versa? Any questions on chapter 4? Okay, so we'll head over to lab. We'll get started right at 12 with those 2 pre-quizzes. One will be on lab 5. The other one will combine lab 6 and 8, as was already on the schedule. Okay, meet you guys over there. I'm not really going to talk at all about lab 5 today. I'm going to put together that handout for you whenever it's ready. I will post it on Canvas. I will also try to print a hard copy for everyone. And then I'll talk to you a little bit about what that lab would have been all about, just so you guys feel hopefully a bit more settled about it, even though you didn't get a chance to actually work on it. Today what we're going to do is focus all of our attention on lab exercise 6, which is called Molecules of Life. In my mind, this lab exercise is married to chapter 3 from lecture, which was our chapter about organic macromolecules. Right? In that chapter, we talked about 4 big categories of organic macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So from lecture, you already know the structure and function of all of those molecules. What this lab is all about, sorry, forgot to unlock it. Sorry about that. What this lab is all about is basically how to detect the presence of those kinds of molecules in a solution in the laboratory. We're only going to look at 3 of the 4 categories. We're not going to look at how to detect DNA or RNA, so nucleic acids are like off the table. Our focus in this lab is on carbohydrates, proteins, and lipids. Before I go any further, I want to have somebody sitting by the hot plates, plug it in, and turn it on, like all the way up. That way, while I'm explaining the rest of the lab, the water can start heating up, and it will hopefully be hot by the time you need it. If it starts boiling rapidly, you can turn the heat down a little bit at that point, but for now, crank it all the way up so we can get them going as quickly as possible. Okay, so this is a lab where you guys are going to work in groups. Each group will have 4 or 5 people. You can't do more than 5. You could probably get away with 3, but 4 to 5 is the perfect number, so try to get a group of people around you, whatever, 4 to 5 people. What I want to do is remind you that always you're going to follow the directions in your lab book carefully, but I also want to give you an overview, kind of what you're going to be doing, what to look out for, all of that. So this lab is divided into 4 different parts, okay? Part A is going to help you figure out how to test for 2 different kinds of carbohydrates. You're going to do a test, it's part A, method number 1, for starch. Remember guys, starch is a big polysaccharide used by plants for energy. We learned 2 in lecture, amylose and amylopectin. Huge polysaccharide. And then in A2, you're going to test for a smaller sugar like glucose. So you're actually doing 2 different tests using 2 different reagents, which is just a chemical you're going to add to your test to pick up on 2 different kinds of sugars, okay? In part B, you're going to test for proteins. In part C, you're going to test for lipids. And then in part D, if you want like a little story to go along with this lab, we have an unknown solution over here. Ooh, yes, mysterious unknown solution. Your job by the end of class is to figure out what's in here. It's going to be some combination of starch, sugar, protein, and lipid. I'm going to just point out right away, if I hold this up, do you guys see there's a separation here? Yeah. So if you had to take just a wild guess, do you think there's a lipid in here? Yeah, good guess, okay? This is why I'm pointing that out. There's a note on here. Before you take a sample of the unknown, you have to shake it. Because if it's separated like that and you only get the top layer or the bottom layer, you're going to get the wrong answer for a couple of your tests. So make sure it's well mixed, okay? So overall goal of the lab is to figure out what's in there. But if you've never done a test for starch, sugar, protein, or lipid, even if you did the test perfectly, the results would be meaningless to you. That's really what parts A through C are about, kind of establishing how the tests work, so you can see what a positive result looks like, what a negative result looks like, and then you'll know how to interpret the results you get from the unknown. That whole idea of a positive control and a negative control is actually the first thing I want to talk to you about, because this is how the whole lab is set up. I'm going to talk about the starch test, which is the first one in the book as my example. I'm picking on this one because it's a test you're going to do a lot over the next several weeks. We're going to take advantage of this test in multiple labs in multiple ways. So if you sort of wanted to follow along, I'm walking you through part A1. What that's going to tell you to do is essentially get 2 test tubes. One of them is going to become your negative control, and one of them is going to become your positive control. A negative control is essentially a solution that you know for sure does not have the substance in it that you're looking for. We're going to use the same thing for our negative control for all of these tests. You're going to use water. You're going to use distilled water. We have little squeeze bottles of distilled water off to the side. Each sink also has a tap of distilled water off to the side. I think you can get more control by using the squeeze bottles that are where I'll show you in a minute. Wherever your source of distilled water is, distilled water does not have starch, sugar, protein, or lipid in it. We know this. So we know that no matter what we're testing for, it should not be in there, okay? On the other hand, your positive control is a solution that you know for sure does have the substance you're testing for, right? So you're going to have different positive controls. For the starch test, you're using a solution of starch, which we have purposely prepared with starch in it. Now what you're going to do for the starch test specifically is you're going to add a reagent called iodine or iodide reagent. It is in a little brown dropper bottle kind of spread out along the benches. I think there are 4 sets of reagent bottles, 2 up front and 2 in the back, 1 on each bench in each spot. Read the labels carefully, of course. What you're going to do is carefully following the instructions in your book. You're going to add some water to this tube, add some starch to this tube, and then to both you're going to add iodine, which is the reagent to detect the presence of starch. When you add iodine to just plain water, and I believe this is in your book, so I'm not like giving it away, you're going to see that solution of water will turn like a amber color, like yellowish orangish kind of a color. That tells you that whenever you see that color, it means there's no starch present in your solution. On the other hand, when you add iodine to this solution of starch, it's going to turn a dark bluish black kind of a color. That darker color indicates the presence of starch to you. Most of these tests are based on a color change like that, where it's one color for a no and a very obviously different color for a yes. There's one test that's not based on color change. I'll tell you more about that one in just a minute. So if you look at your data sheet for part A on page 49, just so you kind of get used to what you're going to have to record here. For the starch test, you're going to fill in the reagent, iodine, and these are the 2 tubes you will have set up when you've done this, a negative and a positive. Honestly, you could fill in these answers right now if you wanted to, but you're going to do this test for yourself to make sure you see the colors you're supposed to see. At that point, you know how the test works. So at that point, you're going to grab a 3rd tube. What do you think you're going to put in the 3rd tube? The unknown. The one you actually want to figure out if it has starch or not. You're going to add some, clearly I was not looking when I was writing. You add your unknown to it, then you add iodine, and your question is, what color does that turn? If it turns an orangey kind of a color, then you would know there's no starch in it. But if it turns a dark bluish black color when you add iodine, then you know it did that because there is starch there. In other words, the importance of doing the positive and negative control, once again, is it allows you to correctly interpret the results from the test with the unknown. Okay, so again, you're doing 4 different tests. And your book walks you through details of how to set up all of them. So here come really like my big tips for how to get this to go smoothly. You have to read and follow the directions in the book carefully. You have to measure carefully. You have to mix your test tubes thoroughly. Each group of 4 to 5 people is going to need a total of 15 test tubes. So what I would do to minimize the backup back here, send 1 person from your group back here to grab maybe a couple racks, like 2 for your group to share, and 15 tubes and take them back to your station. That's where you want to work on labeling your tubes. You must label all of your tubes carefully for a couple of reasons. Number 1, you want to make sure you know what was what. Now, if you only have 2 tubes, a negative and a positive, it might seem like not a big deal, like I'll remember which is which. But if you had them in your test tube rack and someone in your group took your rack and brought it back and set it down the other way in front of you, do you still know which is which? Always label your tubes. How are you labeling these? With those little crayons that are in your black trays, the China markers or wax crayons, whatever you want to call them. They write really nicely on glassware. If you write on the glassware, part of your cleanup is to remove that mark when you're done. Those marks that you write on with the crayon, they come off really easily just with a dry paper towel. Just wipe right off at the end, okay? The other thing you're going to label on your tubes, this lab is kind of weird in that we're measuring volumes. You guys know metric unit for volume is like a liter or a milliliter. But if you look at the method section in your book, it's telling you centimeters of stuff. Add 2 centimeters of this, add 1 centimeter of that. I think honestly the lab is written that way to try to expedite the process for you. Here's what I recommend you do. Remember to use the centimeter side. What I would do is, regardless of how many tubes you're going to be doing, take one of them, hold it to the 0 line, and if you're measuring 1 centimeter or 2, make a little tick mark on the bottom. Then take the rest of however many tubes you're doing and just trace the line across. Make sure you have pre-marked the amounts that you need, because the point here, I guess, is that it is much easier to fill to a line than it is to try to hold a ruler with the test tube while you're adding something and stop at just the right place. It makes your life a lot easier if you kind of pre-mark off where you need to fill to. And read your whole method section before you do this. Some of the tests you have to fill to 1 centimeter with one thing and then one with something else. So tick off every mark you have to fill it to and then follow your directions as you fill it. The other big point I want to make about your test tubes is that anytime you add something to them, you have to mix it thoroughly. I'm just going to put some water in here to make a point real quick. You're not adding tap water to any of yours. There's some water. When I say you should mix it, I see people doing this. Like, that does nothing. Nothing. That's not mixing thoroughly. Hold your tube on an angle, make a peace sign, and strike it on like a downward angle. See how much better that's getting mixed? I'm hitting that with a pretty decent amount of force. You're not going to break it. I've yet to see, hopefully I didn't just jinx us, I've yet to see someone break a tube by doing that, okay? So you have to remember to mix your tubes. In today's lab, as well as on Wednesdays, I'll repeat this then, leaving 1 drop of undisturbed solution on the bottom can change your results. So doing this is not going to cut it, okay? Every time you add something new to your tube, mix your tube thoroughly, okay? When you're done for the day, I had water, so I dumped it down the drain. Are you going to have plain water, only water? Nope. You don't dump anything down the drain. There's a waste container up on the front desk where I usually stand. All of your waste goes into that waste container. Then you will wash. I would wipe the marks off the outside first and then wash with soapy water, rinse your test tubes. There's soapy water already in the gray trays, or if you want to, you can get a little scoop of glassware soap. There are some test tube brushes at each sink. Rinse them out, and then you're going to leave all of your tubes upside down so they can drip dry before the next class in this rack in the back. Let's talk about time management. Best way to get this done is divide and conquer within your group, right? So if I've got a group of 4 people, we've got 4 different tests to do: starch, sugar, protein, lipid. One person take each test and do it. And then at the end, when everyone's done, you regroup. If I had the starch test, I explain to my group how the test works. I show them the negative. I show them the positive. We look at the unknown together and discuss what that result means, right? The key thing to remember is that even if you're dividing and conquering in your group, you personally will still be held responsible for knowing the results of all of these tests. So do not dump any of your tubes until every single person in your group has seen the tubes and has recorded the results together as a group. Speaking of the results, we will get back together as a class to go over these results at the end, because I want to make sure everyone saw the right results in the right tube. Some of these tests can be a little finicky, and you want to make sure you know what you're doing and how they work. So I want to make sure everybody saw the right thing at the end. Just as a heads up, in your group, so everyone's on the same page, the sugar test, part A2, that test takes the longest because that person actually has more tubes than everyone else. Everyone else will kind of have like 3 tubes, the positive, the negative, and the unknown. But for the sugar test, it's the only test we're doing that's also slightly quantitative, meaning it's not just a yes or a no. You'll get different colors based on how much sugar is present. And we want you guys to see kind of like all across the spectrum, from a little tiny bit of sugar to a lot of sugar, what the different colors can look like. So the person doing the sugar test has 5 positive controls, 4 or 5. I think 4 positives plus the negative, plus the unknown. That test also takes the longest because it's the only test you have to boil before you can interpret it. The rest are like add your reagent and look at it. This one you have to add your reagent to whatever solution you're testing. You boil it for 3 minutes. Only upon boiling will the color change. So once the 3-minute boil is over, then you take your tubes carefully out of the boiling water and interpret the results. Putting them in is not so bad because the tubes will be cold when you put them in. When you take them out, you obviously have to be really careful not to burn yourself. There are a bunch of little test tube holders by each hot plate. You just squeeze on the handles, it'll open this up. You can then, grab the tube with it and move it. Still be careful. If you're worried at all about it, call me over, I'll help you take it out of the hot water. The other thing I want to mention is that if you look at the setup of the lab, All of the unknown, like how to test the unknown for everything, is separated into part D, which is fine. I get it. It makes sense because in parts A through C, you learn the tests and how to do them. And then in D, you all test the unknown to apply it. I just think in terms of streamlining our activities today, chop part D up. Meaning, if I'm doing the starch test, I'm going to just add one more tube and test the unknown for starch. If I'm doing the protein test in part B, I'm going to do the part of part D that's testing the unknown for protein. I'm going to add one more tube and test the unknown. I think you'll see what I mean as you look through it, but it goes a lot more smoothly if the person who knows that test tests the unknown for that thing. Instead of putting one person on part D and they have to run around doing all these different tests they don't know how to do, chop up part D so each person is testing the unknown for their molecule. Any questions? Okay, so remember, label your tubes, mix them thoroughly, follow the directions carefully, and we'll regroup at the end. Some of these tests are very quick. I know it sounds like kind of a lot. Some of them you'll be done in 5 minutes. Whoever finishes first, like the starch people and the protein people, right away start helping your sugar person. Because that test takes longer, that person will still be working. See how you can help them to get your group done faster. Okay. Any questions before I set you free to get working? Okay, so get in groups of 4 to 5. Send someone back here to get 15 tubes for your group, and then start to get working on it.
Good luck with today's lesson.